This is the lecture on muscle. Okay, so muscle. This lecture is based on part of chapter 3 from Martini's, uh, 3.5, and chapter 9 on skeletal muscle, a little bit from chapter 21.3 cardiac muscle and 25.1 digestive muscle. Here are the learning objectives written out in detail, and here are the drawing assignments. Here is a sarcomere. Here is a muscle fiber or muscle cell. Here is a cardiac muscle cell and cardiac muscle tissue. And here is a smooth muscle cell. Okay, muscle. There are 3 types of muscle tissue: skeletal, cardiac, and smooth. All 3 types of muscle tissue share 4 characteristics. They contract or shorten. That's the only action that they can do other than relaxing and stopping contraction and lengthening. They respond to stimulation by the nervous system and hormones. They can contract from a range of resting lengths. We really won't discuss that further, and they can return to their original length after they contract, which we won't discuss much either. There are more than 700 skeletal muscles. Skeletal muscles are attached either directly or indirectly to bones. Sometimes they are attached to connective tissue or skin. They pull on the bones or the skin to create movement, and they always pull on bones or skin. They never push. That's not something that they can do. They always get shorter when they contract, which I already mentioned in the prior slide, but it's very helpful to memorize because it will help you to understand everything about the actions of the different muscles when we get to them. Skeletal muscles are under voluntary control in some instances and under involuntary control in other instances. This is a major difference from smooth muscle and cardiac muscle, which are always under involuntary control. Skeletal muscle has many functions. It moves bones to create movements. It maintains body posture and position. It supports and protects organs. It controls entrances and exits to the body. And it helps to maintain body temperature because the act of contracting uses energy. Some energy is lost as heat, and the heat warms the body. And here is a hardworking horse and a hardworking cowboy. Poor horse, doesn't look very happy. Okay, so names and prefixes used for muscles. Skeletal muscles cells are called muscle fibers. Okay, now I know that we learned about collagen fibers, reticular fibers, elastic fibers, and we were talking about proteins that were, small. They were formed of subunits that were synthesized in cells and then were secreted from the cells, and then the subunits joined up and became longer fibers. But in this case, we're not using the word fibers to mean a protein structure. We're using the word fibers to mean cells. Okay, so that is really important to get in your mind. I will try to consistently call muscle cells fibers so that you get used to it and not sometimes call them muscle cells, but I'm sure I will. But in any case, try to remember that because it will really make your life in this lecture and in the next 3 lectures easier. Okay, so sometimes the names for structures associated with muscle fibers contain the word myo, meaning muscle, or sarco, meaning flesh. A complex structure inside the fiber or cell is called a myofibril. The stem cells in skeletal muscle cells are called myosatellite cells. The cell membrane is called the sarcolemma or flesh husk, and cytoplasm is called sarcoplasm. So a lot of vocabulary to learn. Skeletal muscle cells are similar to a typical cell in some ways, but have many things that are different from a typical, little cell as we learned cell in biology. During the development of an embryo, hundreds of myoblast cells fuse to form a single skeletal muscle cell. Therefore, the muscle cells have hundreds of nuclei. because they're made of hundreds of cells that have fused. That's the last we'll see of myoblasts. Okay, they belong in development. They're not part of muscles after that. The nuclei of the muscle fiber is under the sarcolemma rather than in the center of the cell. Muscle fibers are long, whereas an average cell is 0.01 centimeters long. An average muscle fiber is 2.5 centimeters long. So that's 250 times longer. A single muscle fiber in a leg muscle might be 30 centimeters or 12 inches long. So that is crazy, 3,000 times as long as an average cell. Skeletal muscle fibers have a complex intracellular structure. They contain hundreds to thousands of myofibrils, which are complex protein structures that extend the entire length of the cell. The function of myofibrils is to contract or shorten and therefore shorten the whole cell. Myofibrils are composed of protein structures called sarcomeres that repeat along their entire length. It is actually the individual sarcomeres that create the contraction or the shortening of the myofibril. Right? The combined action of all of these sarcomeres is what contracts the myofibril. Myofibrils and sarcomeres are shaped like barrels or like long rods. Okay, they're not a flat structure. They're like long. Let me put are shaped like rods. I think barrels is maybe less familiar. Okay, sarcomeres extend from a Z line to a Z line. The Z lines are protein structures. Z lines attach one sarcomere to another in a row, like cars in a train. So if we go back to this picture, no, none of these show it great. All right, the sarcomeres are made of thick and thin filaments. Thick and thin filaments are themselves complex structures that we're going to learn in detail. The thick and thin filaments are composed of multiple types of proteins. The sarcomere has a middle or an M line that is also a protein structure in the center of the sarcomere that stabilizes thick filaments. Thin filaments are attached to Z lines directly. Thick filaments are attached to Z lines through tightened proteins that pass through the center of the thick filament. Actin in thin filaments and myosin in thick filaments are proteins that bind to each other and cause contraction of muscle. Actin proteins are found in two twisted rows in the thin filaments. Each actin has an active site where a myosin protein in the thick filament can bind to create a contraction. Tropomyosin and troponin cover the active sites of actin, and they must move for the actin and the myosin to bind to each other. Thick filaments are composed of a bundle of myosin proteins around a strand of titin. A single myosin protein has a long tail and two globular heads. Thin and thick filaments overlap in two zones of overlap on either side of the M line. It is only here that thick and thin filaments can interact with each other. Around each myofibril, there is a sarcoplasmic reticulum, which is an organelle that stores and releases calcium ions. Calcium ions are needed to trigger muscle contractions. Transverse T-tubules are deep enfoldings of the sarcolemma, or cell membrane, that extend into the cytoplasm and form a network of small tubes. The T-tubules surround the sarcoplasmic reticulum and so surround the myofibrils because the sarcoplasmic reticulum is around the myofibrils. The sarcolemma and T-tubules conduct electric impulses to the sarcoplasmic reticulum. This triggers the release of calcium ions from the sarcoplasmic reticulum, and the calcium triggers contraction of the sarcomere. Motor neurons, a type of nervous tissue cell, are found in the spinal cord, and they have axons or long extensions of the neuron that's in the spinal cord that carry electrical impulses. The axons branch into fine telodendron, or ends of branches, and the end of a telodendron has a structure called the axon terminal, at the very, very end. An axon terminal comes very close to a single muscle fiber at a neuromuscular junction, abbreviated NMJ. However, not said NMJ, rather said neuromuscular junction. The neuromuscular junction is usually in the middle of the muscle fiber, however long it is. The neuromuscular junction consists of an axon terminal and a motor end plate, a special area of the sarcolemma of the muscle fiber. The axon terminal and motor end plate are separated by a tiny space called the synaptic cleft. A synapse is a place where an axon passes an electron impulse to another cell, usually by turning it into a chemical called a neurotransmitter. A neuromuscular junction is a type of synapse. At the axon terminal of the neuromuscular junction, the electrical impulse causes release of a neurotransmitter called acetylcholine into the synaptic cleft. The motor end plate contains receptors that bind the acetylcholine, and binding of the acetylcholine to the receptors creates an electric charge in the sarcolemma. The electric charge spreads over the entire sarcolemma and also down the T tubules. The electric charge spreads to the sarcoplasmic reticulum and triggers release of the calcium ions. The calcium ions are directly released onto the sarcomeres. In the